So hi everyone. I hope you can hear me. You can hear me all well. I'm really honored to be here and really thankful to ANET for inviting me to give this talk. And I'm really glad, and I think it's really important to that the clinicians and the parent and patient community we speak together, especially understanding that the difficult times that Israel is going through at the time. On top of that, the having to care for all your children with neuroblastoma. So I'll try to give an overview of the future, trying to have. We always have a mixed vision that there has been a lot of progress over the past few years, and with in these current times, we're really making progress very rapidly with lots of advances, and neuroblastoma is a particularly important childhood cancer where we're witnessing significant advances. But on the other hand, there's a lot to do, a lot still to do. And if we are going to be the kind of a case of or an example for other types of pediatric tumors, there are still many challenges until we really improve the outcomes and reduce the suffering of our patients. So I'll start with the presentation. I hope you can see the presentation in full mode and everything. So I'll here have some a conflict of interest statement, and I will start with the talk. I will aim to give a bit of an overview. I know that Dr. Ash, that we work really closely together in Europe and in Siopen, will give you an overview of the current treatment options. So I'll try to focus on the future and try to cover a bit of the what are the new therapies that are coming for neuroblastoma that are becoming available, or just we've had results recently. But also I want to show how we are developing the new drugs, because we have to be researching a lot in the laboratory so that many drugs enter the pediatric trials, and then these trials move into neuroblastoma, and then they move from what we call phase 1 to phase 2 and to phase 3 trials, so that in the end we get to improving the treatment for all patients across the globe. And that we will do with medicines, with chemical medicines, but also with new therapies like immunotherapies, like cell therapies, and others, all in combination. We'll also want to touch a bit on the global landscape and the regulatory initiatives. So I'll try to just briefly mention or explain that as you are aware, the treatment for children with high-risk neuroblastoma is very complex and long. There are children with what we call low risk or intermediate risk that need no treatment or minimal treatment or less treatment, but for high-risk patients, those that have metastasis or amplification of the MECAN gene, we do need to give very intensive treatment, and that has an initial part that we call induction with chemotherapy and surgery, then consolidation with stem cell transplantation after high-dose chemotherapy and radiotherapy to the tumor, and then what we call post-consolidation or maintenance, which is the treatment with anti-GD2 immunotherapy. And first, we need to understand that the treatment often can fail or doesn't work, all this frontline treatment. And the first way that it does not work is when we give the induction chemotherapy to reduce the metastasis, to get rid of all the metastasis, and even some of them may respond, but not all of them. And this is what we call refractory disease. So the patient, we expect a very good response, that all the metastases vanish, and not all of them do it, and that's what it's called, refractory disease. And these patients need additional treatment, and if there is a response with the additional treatment, then they can go back to consolidation and post-consolidation to the rest of the treatment. But the biggest problem is what we call relapse or progression. So at any point during treatment or during follow-up, the treatment, the tumor may start growing again, despite the treatment that we are giving or we have given. And that's what we call progressive or relapse disease. And that's the biggest challenge to face now, because it is the tumors have become a lot more resistant and it's harder to treat the patients. So I wanted to touch on a few of the results that we've seen in the last 2, 3, 4 years that are now becoming options quite common for patients with relapsed neuroblastoma, and hopefully soon they will become common for patients at the beginning, at just when they are diagnosed, because we have the philosophy that treatments that are effective at the time of relapse, we should try to test them and give them upfront, so because we want to cure as many patients as we can at the beginning without sparing having to have a relapse for the patients. So chemoimmunotherapy is the combination of any chemotherapy with anti-GD2 chemotherapy. And there are many regimens and different anti-GD2 antibodies that have tested. There is one anti-GD2 antibody that is used in North America called dinutuximab, another one that is used in Europe and the rest of the world called dinutuximab beta. There's another one which is more limited. Its use that is called naxitamab. There's another one that was developed by the St. Jude Hospital. But basically, all of them have the same target. And in combination with chemotherapy, all of them have similar results. We saw a few years ago a trial from the Children's Oncology Group in North America that showed that with the combination of this dinutuximab with chemotherapy, they had a good response rate, which is the proportion of patients where the tumors shrink, that reduce, and also good progression-free survival. And these type of curves, I'm sure you'll start to see them or to be familiar in many of the publications. As this survival curve just shows the time on the horizontal axis, and at the beginning of 100 patients that start, it shows the proportion of patients that remain without a progression. So the longer the time, we see that with this type of treatment, some patients can get rid of the relapse, but what happens in many patients is that the relapse is delayed, which is still an important effect, but we would like that to be maximized. And we did a similar trial with dinutuximab beta and a similar chemotherapy in Europe, and showed again a similar figures, showing an improved survival. The other important milestone is the identification of the ALK gene, which is altered. This gene is affected in about 10-15% of the children with neuroblastoma. They may have a mutation, which is one type of alteration in the sequence of the genes, or amplification, which is many copies of the same gene. What we know is when these aberrations are present, the tumor is very dependent on this ALK gene, and that's why, because there are ALK inhibitors that have been developed for other adult cancers like lung cancer, we're trying to use these agents to improve the results in neuroblastoma. And for example, the North American consortium called NANT has done a phase 1 trial with lorlatinib, that is one of the most potent ALK inhibitor, and has shown good results in patients, in children and adolescents and adults. A bit better, the results are better in adults, although they're not very frequent, but they respond very well to lorlatinib when they have these ALK mutations, and also in combination with chemotherapy. And again, the same in Europe. We have done trials with ceritinib, which is another ALK inhibitor, again showing that it doesn't benefit all the patients, but some of the patients really benefit for quite a long time. So the plan is now to incorporate these ALK inhibitors upfront, and currently they are only used at the time of relapse. And the third kind of important news in the field is that from last year, the group of Rome of Professor Franco Locatelli reported a CAR T against this GD2. So this GD2 is a molecule that is in the surface of the neuroblastoma cells, and is a very good target for neuroblastomas. So they developed a CAR T, which is a specific type of therapy where your own lymphocytes are changed so that they attack the cells that express anti-GD2. So they extract the blood cells from the patient. They're processed in a manufacturing facility so that they attack this GD2, and then they're infused back. So this was a very important milestone in the field of pediatric cancer, because it's the first time that a CAR T has shown very clear activity, signs of activity, in a solid tumor. The CAR Ts have been successful in leukemia, but they haven't been successful in other solid tumors. So this is the first time that a solid tumor is responding. And this is really a very important milestone. A significant proportion of the patients responded, a significant proportion of patients remained without a relapse during treatment, and particularly those that had less disease burden. So those that had already responded to something then benefited more from the CAR T. So this is a very promising initial step, and now what we have to do is to make that available across a multi-center setting, so in different countries and different centers, to find which are the patients that really benefit from these CAR T trials, because we already know that some of the patients that have a lot of disease, for example, at one point, a lot of disease burden, will not benefit, and then should go for other treatments. And then at the same time, there are other groups in London, in Berlin, in Utrecht, and in different centers in North America as well, developing other CAR T against GD2 and against other pediatric targets that also hold significant promise. And I think that, together with other studies, has made that when a patient has relapsed neuroblastoma, there are many options, and that's the negative side is that still a lot of patients with neuroblastoma experience relapse, and the positive side is that we have multiple options to consider. And I will not go through all of them, but I think it's better to be in the position where we have more options, including some more conventional options and more innovative ones, with combinations with molecular radiotherapy, with immunotherapy, all different combinations to provide access for the patients. But how do we move forward from here? We have a number of challenges that we keep working to overcome. The first one is that we do need faster and better access to the early phase clinical trials. The trials are the way that we have to make advances, because they're the only way to evaluate that a new treatment is safe, that a new treatment works, and to show and convince the rest of the world that a particular treatment is working, and it should be adopted and be made available across the globe. So one challenge is that because the neuroblastoma patients are not so frequent, we do need, the faster the clinical trials open and the faster they are completed, the faster we will get the responses. And if a combination or a treatment is not working, then we will know earlier and be able to test the next one. And if it works, then we will be able to benefit more patients more rapidly. The second challenge is that we do continue to facilitate the transition from preclinical work from the laboratory into phase 1 trial, the first time that the drugs are tested in children, and then into phase 2 and subsequent developed. And these, now it's becoming even more complex because now some of the therapies that we're developing, they're only for a small group, and that's the example of the ALK inhibitors, that they're only useful for 10% or 15 or 20% of the patients, and not all of them. And also because the combinations, we do need to give the new therapies in combination to make them more effective, or therapies like the CAR Ts are more complex to develop and to administer. We are also working extensively to harmonize the way that we measure the results and that we measure the outcomes across the globe. And this means using criteria that the whole international community accepts and understands, whether it can compare whether one drug works better than other. And also importantly, incorporating patient-reported outcomes, not just the measurement of the tumor, but whether the patient was deriving benefit. They had better quality of life. They had less pain. These are all important points to decide whether a medicine is working or not. And finally, there is a lot of global work and international work in neuroblastoma, but we still need to improve it. And then in the end, to make available the drugs that are effective, to make sure that they're available across the globe as rapidly as possible. And here, in this point, we still have significant challenges, and we do have significant challenges in North America, Europe, and the Australia and the Western world, let alone to make these treatments available for the whole world. So, what's coming next? I think it's reassuring that there's a lot of clinical trial networks working together. In North America, there's the Children's Oncology Group, takes care of the frontline clinical trial and the trials for first relapse, as does the Siopen in Europe. And then there are smaller consortia that take care about early phase clinical trials, the phase 1 and phase 2 clinical trials like NANT in North America or ITCC in Europe. And then there is clinical trials that the pharmaceutical companies start testing their medicines and developing better combinations. And also there are institutions that take forward their own clinical trials and also contribute to the field and move so that we move forward. So just as a start, what do we do with all of this in Siopen? Well, that's the example that the current high-risk neuroblastoma 2 trial is embracing some of these advances. And for example, we are incorporating chemoimmunotherapy. So chemotherapy with anti-GD2 immunotherapy is now started to be given in the trial has been amended to give chemoimmunotherapy for patients that don't respond so well, so those that have refractory disease. And then we are also starting what we call a pilot trial to give it to every patient at the beginning, from the beginning, to give the induction chemotherapy that we're giving, that is called rapid COGIT, that is very intense and rapid chemotherapy, to add the anti-GD2 to that. We are also working to incorporate lorlatinib, the ALK inhibitor, for patients that have ALK-positive disease right from the beginning. But again, here, because we want to ensure that if it works, it will be used in the future with no problem. We want to do a randomized trial to show a clear benefit, to be able to show that there is a clear benefit on adding lorlatinib, that the benefits outweigh the risks, so that we can convince the regulatory agencies and the funders and the healthcare systems across the globe that lorlatinib should be incorporated for all ALK-positive patients. There's a new situation with DFMO, or eflornithine, that is an agent that has been recently approved by the FDA, and we understand that will soon be available in Europe, in Israel, and in other countries. But still, we have to make sure that it's approved by the relevant regulators, so that we can use it. If we want it, we can use it without limitations. And at the moment, it's only approved by the FDA and not by the rest of the agencies in the world. And that brings a challenge, because it is available in the US but not in the rest of the world, and we want that gap is, while understandable, is as short as possible. And then in the future, we are already thinking of what would be the next clinical trial, and that would be the high-risk neuroblastoma 3 trial, with the aim of reducing toxicity, because some of the parts of the current treatment are really intense in the short and in the long term, and incorporate new medicines or new therapies, and combine with immunotherapy and with targeted therapies to try to achieve better results with improved survival and less side effects. What is coming for relapsed neuroblastoma? So our North American colleagues have recently finished the recruitment for their clinical trial, ANBL 1821, that tested chemoimmunotherapy plus or minus DFMO. Is a different use than the current use of DFMO. This is given together with the chemotherapy and the immunotherapy, and we're just waiting to hear within the next here the results of the trial. And then in Siopen, we are opening up together with ITCC the Beacon 2 trial, that following on the Beacon trial that we conducted in relapsed and refractory neuroblastoma, we want to open a platform, what we call is a platform trial, which is a trial where you can continue adding more arms as time goes by, because we think it's really, we have to be flexible in this situation. We cannot run very large trials, so it's better to do smaller trials and that are flexible, so that if there's new data on one agent or a combination, we can move forward. And with this, we will have a bigger randomization with arm A, B, and subsequent arms as they are added. At the moment, it's only starting with the arms that we've already tested in the Beacon 1 trial, but we're hoping to include newer arms. And then we will have what we call tier 2, which is smaller cohorts for patients to test newer combinations, to gain some data on the newer combinations that then can be taken into the larger randomization. We have got 16 countries lined up, including Europe, together with including Israel, Australia, New Zealand, so a significant part of the world. And the trial has already recently been submitted to the regulatory authorities. So we hope that we will be able to open it in the rest of the countries by the end of this year or beginning of next year. And we're already working with different pharmaceutical companies, with different investigators from across the globe to find newer combinations with immunotherapy or with targeted agents to improve outcomes. This is just not to show all the clinical trials that the ITCC has in their portfolio, but it's more to show that that we are better in having more options. There are more options for biomarker-driven trials. There are more options of other targeted agents and more options of different immunotherapy combinations. And we have made progress in that there's an increase in the number of trials available in Europe. They are more focused in neuroblastoma. There are more trials in combinations, which we think it's really important because the combinations have a better chance of working than single agents. And there are more drugs that are moving forward in the development that go from phase 1 to phase 2, etc. So we're really hopeful. We are doing an exercise of communication in Siopen that Alba Rubio and Pablo Berlangan are taking care of, trying to give a priority of all these trials that are available to make sure that the clinicians and the patient community know that there are all these trials. And when they are high priority, we put them in green, so that that means that there is a very good rationale for that trial in neuroblastoma, so that patients could travel from other centers within the same country or from other countries if needed. And we're working with ITCC in different initiatives to facilitate the access to early phase clinical trials. I wanted to, as I mentioned, I wanted to touch base on how are the drugs developed? How do we work to have more drugs for neuroblastoma? So this is pretty generic. This slide is pretty generic for any type of cancer. So there is a phase that we call preclinical, which happens in the laboratory. First, we look at the tumors to see what are their differences from the normal cells and that we could target. Then we try to discover drugs, to discover the medicines that would hit these vulnerabilities, these hallmarks of the cancer. And then we do testing of drugs in the laboratory that can be in cells and can be in animals as well. So when a drug is successful, then it's moved to phase 1, what we call is the first that it's given to humans or to children in our pediatric oncology, where we want to confirm that the dose is right and the medicines are safe. And then we move to phase 2, which are smaller studies, but already specific, and then we move to the larger phase 3 trials, which are necessary to prove that the new treatment is better than the existing treatment, and that facilitates that the regulators approve the treatment, and then it's available for clinical use. And there are many barriers, many challenges. We have an improved knowledge of neuroblastoma, but still there's a lot to know. There is, in pediatric cancers and in neuroblastoma, the resources dedicated to drug discovery, to finding new drugs, are limited, and there are some targets that are pretty exclusive for pediatric cancers, and MECAN, the MECAN gene is one of the examples. So there's not enough efforts in discovering new drugs against a target like MECAN. And then preclinical testing is still imperfect, and we need to improve how to use it. And then it takes really long time to take the drugs into phase 1, to have access to the drugs for pediatric trials. It takes a long time to open the trials and to conduct them. And then for the phase 3 trials, we cannot do too many of them. There's only 1 or 2 that can happen at a time. And then once the drugs are approved by FDA or EMA in Europe, still the drugs take some time to be available across the globe. And where are we working at the moment? What's going to be the next generation of clinical trials? Well, we now have a good understanding of the priority targets. And of course, MECAN is one, ALK is another one. TERT is related to the telomerase maintenance mechanisms that for a particular group of patients, they make them more aggressive. And there's the RAS-MEK pathway, the ATRX. So we know a number of vulnerabilities of the tumor that we are trying to target. For some of them, there are drugs. For some others, there aren't drugs yet. And we are working for that. And that's more on the molecular level at the cell, to find these vulnerabilities in the neuroblastoma cells. But also, we are trying to develop combinations of these different novel targeted agents. We're developing combinations of the new therapies with chemotherapy, also with immunotherapy. And then in immunotherapy, there's also a revolution in anti-cancer drug development. That is, now there are now more anti-GD2 molecules, and we hope that new anti-GD2s may work better, or may work better in combination, like the example with chemotherapy. But also there are other ways to attack this GD2, the target GD2. There's something called ADCs, which is antibody drug conjugates, that it's a molecule, a quite complex molecule that carries some toxic agent, like a type of chemotherapy, and it's just a carrier that then attaches to the neuroblastoma cell and drops the medicine inside the neuroblastoma cell, instead of the conventional way that chemotherapeutics are given, that they have to enter into the cell. And there are ADCs being developed against GD2, and for CAR-T as well. As I mentioned, there's one example already in the clinic, but there are other groups working to develop CAR-Ts for GD2 and for other targets that are exclusive of neuroblastoma, and that we hope that in the next years will become available, and we will have to do the phase 1 or phase 2 trials, and if they're promising, to take them into the front line. And as I mentioned, the last challenge I wanted to touch is on the international collaboration, because this is really important for progress to happen. So at the moment, there is a very close collaboration between the Children's Oncology Group, between Siopen and ITCC, between the different consortia, and we're talking more than ever and trying that the trials that we do contribute to the trials that COG does, and vice versa. We look at the trials that they have, and we try to have coordination so that the advances are as fast as possible, and make the fastest transition from the laboratory into the clinical uses as we can. So here are some examples to illustrate how this is happening. One is the INRG database. This is a database that contains clinical and genomic data from more than 25,000 neuroblastoma patients from the whole world. So the North American group, the European group, the German group, Japan, St. Jude, different groups and hospitals from the world put together all their data. And this is a really valuable resource to try to find new medicines, to try to find different groups that respond to treatment in a different way. And we continue to work to enhance this, but this is an example that has driven that other pediatric cancers also put together the data from across the world. The NDDS initiative is the new drug development strategy initiative that was started by my mentor, Andy Pearson, as was the INRG database as well. So this is an initiative where we put together at regular intervals, every few years, we get together clinicians, researchers, patient advocates, members, people from pharmaceutical companies and regulators, and we review all the data that is available over the past 2, 3, 4 years on which targets and drugs could work for neuroblastoma. And we do a critical review of all what is available. And then we document, we publish what is a priority. And we hope that, and with this, the aim is that the pharmaceutical companies, the regulators, every researcher knows what we think is a priority, which drugs should move from the laboratory into the patients, from the patients into neuroblastoma trials, or which targets don't have a drug and need people working on that. We've already done 3 editions, and the aim is to speed up the drug development. And then I wanted to mention Accelerate as well, because again, that's an international initiative that brings together clinicians, advocates, pharmaceutical companies, and regulators, and aims to bring together the views, because we got to understand that each of the groups may have a different priority or may have a different perspective, and it's only by working together that we can overcome these limitations to make the pediatric drug development work faster. And for example, a pediatric strategy forum is a meeting where all of these people get together and review one pediatric disease or one target and try to help. And sometimes there are many companies wanting to do trials for very few patients. Sometimes there is a disease that is very difficult to treat and no companies working on it. So in the case, this October is going to be the first one dedicated to anti-GD2 strategies. So we will bring together all the clinicians, patient advocates, pharmaceutical companies, and regulators that are interested in developing anti-GD2 therapies, the ones that are already available and the newer ones that are being developed, so that we can get to an agreement of what is a priority beyond the individual needs of the regulators, of the pharmaceutical companies, and we agree on the way forward for developing anti-GD2 therapies. So with this, I would like to conclude highlighting this half optimist and half pessimistic message. We know that the advances in clinical development are happening and they're tangible, but still they're too slow and too few, because we have got the patients right now that are not responding to treatment, and we do need to keep working and to keep working together. Also, that there are key recent advances like the incorporation of chemoimmunotherapy, the development of ALK inhibitors, or the CAR-Ts, as we have seen. And the future will see the development of new molecules, new combinations, targeted therapies, immunotherapies, cell therapies, vehicles, and the improved understanding of the neuroblastoma biology, all of that together will lead to improve outcomes. We continue to foster and to promote international collaboration and multi-stakeholder collaboration, because it's really crucial to ensure that the advances occur and they get to be implemented. They are available across the globe. So with this, I'd like to finish. I'm really thankful to Einat and Share for Life and Shifra, and to all of the people that work together in neuroblastoma across the globe. And I'm really happy to take questions.
Thank you so much for the amazing talk, overview, and future challenges. So, any questions?
Okay. Thank you for the talk. It was very interesting. My question is about right now we are following the disease progression with scans. What do you think about following it with liquid biopsies, and what is the status of using it for determining, making decisions based on the liquid biopsies.
Yeah, so thank you. It's a really fascinating question and a fascinating field that is moving really, really fast. So genomics is advancing so rapidly by days that this is continuously evolving. And I would say that as of today, still internationally, there's no full agreement on using liquid biopsy for following up the patients. That they are useful when we have identified a specific aberration. So if there is an ALK mutation, for example, we do know that following up that ALK mutation during treatment, we can see that it comes down with treatment, and then if the tumor comes back, we will see it coming up again. So it's well sorted, well resolved for some specific genomic aberrations and not for all the patients. For example, in Siopen, we are now starting a project that is called Mona Lisa that will be open in many hospitals, where we will collect the liquid biopsy samples. And one of the challenges is, what do we do with the results? Do we inform the clinicians? Do we inform the families? Will it change what you do? And is it worth changing only by liquid biopsy? So there's still not completely resolved. And also, we don't know when a very detailed sequencing study is done on a liquid biopsy, like in the foundation one, for example, we still don't know how much that represents the whole of the tumor. We don't know. It may be that it does, but at the moment, we prefer to take the treatment decisions based on the biopsy of the tumor or on the scans. But it can already provide useful information when there are known aberrations. And for the rest, we will continue to find out, and we have to collect the data and understand what is the added benefit of the liquid biopsy. But it's definitely an area where we are working.
Thank you. For the clinical. Any other questions? If not, thank you so much.
Thank you very much. I hope you have a very good and productive meeting.
Okay, thank you, Lucas.
Bye-bye.